My research presentation was on the parasite called Toxoplasma Gondii. So the scientific name is Toxoplasmosis Gondii, also known as T. gondii. It is a gram-positive unicellular parasite that has a nucleus and membrane-bound organelles. T. gondii is a Category B pathogen stated by the CDC, which is a second-level threat with low morbidity and mortality rates. It was discovered by 3 scientists working in North Africa and Brazil in 1908. The first scientist's name is Charles Jules Henry Nicole. He was born in 1866 and died in 1936. He was working in Tunisia, North Africa at the time. You can see him on the bottom right here. The next scientist was Alfonso Splendor, who was born in 1871 and died in 1953, and he also worked in Brazil. You can see him right here, to the right, to the left of Charles. Louis Herbert Manceau was born in 1865 and died in 1934. And as you can see, there are no pictures of Louis available. This parasite can be found worldwide during any season. You can also find this parasite on raw and uncooked meats or shellfish that have been contaminated by feces. It is estimated that 22.5 or 40 million Americans may be infected, with the parasite unknowingly. You can acquire this parasite through 3 principal routes, such as ingestion of raw or uncooked infected meats, the ingestion of octocytes, exposure to infected cat litter, and contaminated drinking water or soil. Lastly, newly infected mothers can pass the infection to her unborn fetus through the placenta. This organism was first discovered in a small rodent native to North Africa called the Gondii. The relationship. It is a member of the Eukarya, and more specifically the phylum, which is known as the Apocomplexia taxonomy, and it can infect both invertebrates and vertebrates. And it is closely related to the dinoflagellates, which are right here, which have flagellas that can move and propel him. It is also related to Plasmodium, which is the parasite that causes malaria. There are 3 forms of T. gondii, which the first form is tachyzoite, which is the acute stage, invades and replicates within cells, and radiozytes, which are latent infections present in tissue cysts. And lastly, the sporozytes, which are found in the osteocytes, which are environmentally resistant. Why is this organism so special? This organism does not show symptoms when a healthy individual is infected. Immunocompromised individuals have more of a risk of developing severe symptoms such as fevers, headaches, nausea, lack of coordination, confusion, and seizures, and many other symptoms. After a couple weeks/days, the octocytes sporulate and become infectious. In favorable conditions, such as in warm, moist environments or soil, the parasite can remain infectious for approximately 1 year. This parasite can also not survive in dry, cool climates, and it can be destroyed by heat. Interestingly, this parasite can influence human culture in both males and females. Males who are infected show signs of lower intelligence and decreased rule-conscious behavior. And females who are infected show signs of higher intelligence and increased rule-conscious behaviors. Does this parasite cause disease? If transmitted from a mother to fetus during pregnancy, Toxoplasma can have severe impacts on the unborn child, such as miscarriage, stillborn birth, or congenital toxoplasmosis, which is the abnormal enlargement or smallness of the head. It can also cause eye disease from the result of congenital infection or infection after birth by any of the modes of transmission. You know if you have eye disease by eye pain, sensitivity to light, tearing of the eyes, blurred visions. It can reactivate multiple times and each time causing more damage to the retina and can eventually lead to blindness. People with compromised immune systems may experience severe symptoms. People who acquire HIV infections and who are not previously infected with Toxoplasma are more likely to develop a severe primary infections, stated by the CDC. Immunocompromised people who are infected with Toxoplasma at some point before they became immunosuppressed are at risk for developing a relapse of toxoplasmosis. A person who reactivated toxoplasmosis can experience fever, confusion, headaches, seizures, nausea, and poor coordination, and also many other symptoms. So the life cycle of Toxoplasma gondii is it is ingested by a host, then the sporocytes invade almost every kind of nucleated cell, and then begin multiplying asexually by endogamy. The sporocytes are now called tachyocytes, which multiply very quickly, forming pseudocysts, which burst open to release more tachyocytes. Now, the tachyocytes are called radiocytes, which multiply more slow, and the termination of bursting pseudocysts. Finally, they form shelled cysts to hide in the host tissues until a second host ingests the intermediate host. So lastly, we have survival. Establishing a successful invasion, T. gondii needs a sequential secretion of micronomy and repertory neck proteins, also known as RON, first to mediate the invasion. Microny is basically the mouth or the anus of the parasite that secretes RON2, which is the chemical protein that allows them to break down the cells and insert themselves into the nucleus. T. gondii receives its nutrition directly from its host body, living in the intestines and absorbing the nutrients directly. The parasite can also live off the components of the other cells, burrowing inside them and directly absorbing the nutrients in the cells to survive. Further research. As of now, there are no currently vaccines available. Human studies have revealed a range of subtle behavioral alterations associated with toxoplasma, many of which may be comparable to the infected rodent intermediate hosts, such as increased activity, decreased reaction times, and altered personalities. Latent Toxoplasma gondii infections in the human host may be associated with some forms of affective and neurological disorders, such as Parkinson's disease, Alzheimer's, bipolar disorder, and even suicide. Here are my resources. Thank you.